need energy. There's no time to wait, so they make ATP from creatine phosphate. So, for a number of reasons, you wouldn't want to be like your cells stocking up on ATP, so like our energy money. You'd have like reactions just like going off wildly in your cells, and other problems. But your muscles need to get this ATP really quickly. And so, in order to do this, instead of stocking up on ATP, they stock up on creatine phosphate. And then, when they need energy, they can transfer the phosphate from creatine to phosphate onto ADP to make ATP, and now you've got ATP without having to store up a bunch of the ATP. And this, what's really cool is that this whole process is like regulated by levels of ATP and ADP, and it's all really cool. And so let's talk about it.
ATP is often referred to as like the universal cellular currency, or like I think of it as like kind of like energy money. And the reason for this is that ATP, so adenosine triphosphate, those triphosphates, so phosphates are these phosphorus surrounded by oxygens, and they have these negative charge. If you stick negative charges close together, it's kind of like you're clamping a spring. So we call these bonds like high energy because they have high potential energy, meaning that if you were to break these up, then you would get a lot of energy in return, kind of thing. So it's kind of like the equivalent of like kinetic potential energy, where if you had like a ball at the top of a hill, in this case, we have this chemical potential energy where if because these bonds, it's so like hard to keep these negative charge groups together. If you were to relieve that tension, you unclamp the spring. Think about if you had like something at the end of the spring, it would like fly off. But in this case, we can couple this release of energy to get reactions to happen that may not be otherwise favorable. And this is often, this ATP is used by enzymes, which are like reaction mediators or speeder uppers, often proteins, sometimes proteins and RNA, sometimes RNA alone. But they kind of help make reactions happen by binding to the substrate, so the thing that's going to get changed, and helping like stabilize awkward transition states and all sorts of stuff. So often reactions, even if they're favorable overall, so even if the products are like better or like happier or more energetic stable than the reactants, you have to go like past like an activation barrier. So if you think about like snapping a stick, when it's like you have to actually bend the stick and stuff in order to break it. And so enzymes can have this like, so reactions have this activation energy, and enzymes can help lower that. And then if you add ATP, now what you're doing is you're actually providing the energy that you need to go. And so often we see ATP used in reactions, both in order to get over the activation energy and or to make reactions happen where the products are not actually that much more favorable than the reactants. So ATP is really awesome, and a bunch of different enzymes can use it. And so it's kind of like a universal currency, kind of like an arcade token. You can stick in a bunch of different starting materials like sugars and fats and proteins, and they can all get broken down. And the energy stored within those molecules can be converted into ATP, which can be stored as energy. But because you have all of these enzymes that are using up this, that can use this ATP, you don't want to just like, you don't want a huge stocks of it just hanging out in your cells, because then you could have all sorts of wild reactions happening when you don't want them. So your cells want to tightly control the amount of ATP. So instead of stocking up on ATP, what your cells do is they stock up on the things that they can use to make ATP. So they stock up on like lipids, so like fats. They stock up on like long fats or long carbohydrates like glycogen, so a bunch of sugar linked together, and then they could break it down when they need in order to get energy. But this breakdown process involves a lot of steps. And so like for if you started with glucose, so if you started with like a sugar, you're gonna have to go through glycolysis, you're gonna have to go through the citric acid cycle, and then oxidative phosphorylation, and you end up with a bunch of ATP, but you have to go through all of this stuff. And so this is taking time. And you can imagine that like if there's a bear coming at you and you need to run, you don't want to be waiting around for all of this to happen. And so you have this kind of conundrum in your muscles. and some other cells where basically you need ATP quickly and you don't want to wait for all that going on. But you still have that same problem where you don't want to have a ton of ATP just building up. And this is where phosphocreatine and creatine comes in. And so what's actually going to happen is, so this molecule creatine we'll talk about, it's going to ATP. If you have a lot of ATP, this creatine kinase, so a kinase is something that transfers this phosphate group from like ATP or another NTP to a molecule. And so we've talked about kinases before, where we into terms of like protein kinases as a post-translational modification. So something that gets added onto a protein after the protein's already like the amino acids, so the protein letters have all been put together. We call it translation. And then things can happen to the protein. Modifications can be put on afterwards. We call these post-translational modifications. And like the main one is phosphorylation. And so we talk about phosphorylation of proteins. So we can have protein kinases, but we can also have small molecule kinases, which are going to phosphorylate small molecules. So things like creatine instead of like big old proteins. And so in this case, we have this enzyme, this creatine kinase. What it's going to do is it's going to take the ATP, take the creatine, and it's going to stick the phosphate group from ATP, this gamma phosphate, the end one, onto this nitrogen of this phosphocreatine. And so now you have ADP, and so you have reduced the levels of your ATP, and so this isn't going to be the same as the ATP. So the ATP is like all these enzymes are specialized to use ATP, and that's why you can use it as like this universal currency, because they all recognize and like bind to it and use it and that sort of thing. And so they're all specialized to use ATP. But now you have your energy stored in phosphocreatine. You have like this potential to make ATP now, but you have it on this creatine, and the enzymes aren't going to recognize this phosphocreatine and be able to use it. However, this reaction is reversible. So although we call it creatine kinase, it can also do the opposite. It can be like an ADP kinase kind of thing. So basically it can go both ways. And the amount of which way it goes. So is it transferring the phosphate from ATP to creatine, or is it transferring it from phosphocreatine to ADP? And that's going to depend on the amount of the ATP present. And so basically you can think about, if you have a lot of products, so if you have a lot of the ATP, then you're going to be driven to this direction to making it phosphocreatine, because you're going to have a lot more of these molecules. And there's going to be a lot less of these molecules, so it's less likely that these are going to react, and more likely that you're going to have these two to go this way. When you have a bunch of ADP, however, now you're going to have your reaction go this way. So you're going to be making more of the normal creatine and ATP. So you're going to be removing the phosphate group from the creatine, from the phosphocreatine to make ATP. So when you have a lot of ATP, you're going to make phosphocreatine. So this is like, dude, we have enough ATP in our muscles. We don't need to be making more. All's good. But now you have when ATP levels are low, so now it's like you need more ATP. And now this is going to make ATP. So it's really cool because it's getting kind of like regulated by the levels of ATP, which is what it's trying to regulate. So you have this like feedback loop. So you might think that this could go on like indefinitely, right? And so you can have it going this way and 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 this way. So why would you ever need to actually eat creatine, right? So it turns out that your this creatine and phosphocreatine, they can like non-enzymatically convert into creatine. So this transfer of the phosphate group, this is happening with the help of creatine kinase. So creatine kinase is an enzyme. This is being enzymatically mediated. So we talked about enzymes before. Remember how they're helping these reactions happen. But enzymes, basically, they're kind of like leading a horse to water. They're not making it drink. And so enzymes aren't actually making the molecules react. They're just making it much more easy for the molecules to react. And there are certain molecular reactions that don't need enzymes at all to help with that, with what they're doing. And so there are non-enzymatic reactions and non-enzymatic processes. And so, for example, this creatine or this phosphocreatine, they can spontaneously and non-enzymatically convert into creatinine. So creatinine is this like ringified version. And so if you look at this nitrogen here, if it were to attack this carbonyl carbon, now you get this creatinine. So this creatinine isn't going to be very helpful. And so it's like a waste product. But what happens is that about 2% of your muscular stores of creatine get like this spontaneous ringification thing to make creatinine. And then this creatinine is going to just like diffuse out of the cells. And then your kidneys are going to filter it into your urine, and you're going to pee it out. And so this is why you need to eat more that's why you need to get creatine in your diet. So basically, the average adult size male has about 120 grams of total creatine, which is like a ton. When we're talking about like tiny little molecules. However, since about 2% of this is going to get this ringification thing, about 2 grams per day needs to be either made from scratch, so our cells can make it, as we'll talk about, or gotten from the diet. So our cells can make it, so it's not like dietarily essential in the sense that you need to get it directly from your diet. We can make it. We actually make it from a couple of amino acids that I'll talk about in a minute. But so sometimes you might have heard of creatinine in terms of like doctors running tests to test creatinine levels. So this is because of the whole kidney thing. So because your kidneys are responsible for helping like filter out this creatinine, this like waste product, if you have problems with your kidneys, then you can have problems with getting rid of this creatinine. And so like if your doctors often measure creatinine and like the urine that's collected over 24 hours, and then they compare it to the amount in the blood to get like the creatinine clearance rate. So it's just basically saying like how well are your kidneys functioning, because how well can they clear this from your pee. And then they often use that to estimate the blood flow through the kidneys, so like the glomerular filtration rate, or the GFR, to get an idea about overall kidney function. So it's not so much that they care about the creatinine, they're just using it as like a marker of this thing should be getting filtered out of the blood. Is it getting filtered out of the blood? And what is the rate of blood flow through the kidneys? So that's why you might have heard the term creatinine, even if you or seen in the context of like medical shows or stuff, and you're like, what the heck is that? So that's where creatinine comes in. So creatinine is different from creatine and different from phosphocreatine. It's this like spontaneous ringification product. It's like this waste product that your kidneys get rid of if they're working properly. So let's talk a little bit more about how our cells actually make creatine and what it actually is. So creatine is actually an amino acid. And so it's not, we've talked mostly about amino acids in the context of being protein letters. Creatine is not found in proteins, but it is an amino acid, and it's made from amino acids. So in order to be an amino acid, something just has to have like an amino group, so an oxygen with hydrogens, and a carboxylic acid group, so like this double bonded C double bonded to an O and then another O. And well, carboxylic acid would have it be the acid form, so we'd have an H here, but it can be it's typically deprotonated, so much more on that in other posts. But the basic idea is that you have an amino group and a carboxylic acid group, and you're called an amino acid. So creatine, if we look at the structure of creatine, we can see that it has amino groups and carboxylic acid groups. So voila, it's an amino acid. But it is not an alpha amino acid, so it's more on that in the amino acid post or whatever, and it's not used to make proteins. It's just like a technical thing that's kind of cool. And so But creatine is made from amino acids that are used to make proteins. And so it is made from two of them, glycine and arginine. But then it's also going to involve a third amino acid, methionine, indirectly, because it's going to be the source of a methyl group. And so a methyl is this like carbon bound to 3 hydrogens. So glycine is the simplest amino acid. And so these amino acids, the protein ones that we're talking about, they have, in addition to that amino group and a carboxylic acid group, they have this unique side chain or R group that sticks off. And in the case of glycine, you just have a hydrogen. And so this is going to be this hydrogen. So that you can see here, this is the backbone we're getting from the glycine, but we're going to modify it. And so there's this enzyme, arginine glycine amidinotransferase, or AGAT, and basically it's going to give arginine's amidino group. So this thingy bob, where you have a carbon and these 2 nitrogens, it's going to transfer this onto glycine to make guanidinoacetic acid. And then because you've removed this amidino group, now what was arginine is now ornithine. And this is you're not quite creatine yet. We still need to add on this methyl group. And this methyl group is going to come from a molecule of S-adenosylmethionine, or SAM. And so basically this enzyme guanidinoacetate methyltransferase is going to move the methyl group from SAM onto this guanidinoacetic acid to make your creatine. And so this methyl group, this is why you need methionine as well, because the methionine is actually going to serve as the source of this methyl group. And so more on that in my post on methionine, how methionine makes methyl moving possible. So in mammals, this first step is going to happen in the kidneys. And then the second step is actually going to happen in the liver. And then what the liver is going to do is it's going to release the creatine it makes into the bloodstream. And then the cells that need it are going to take it in. So only certain cells have the receptors that are needed to let in this creatine. And so these involve cells in your muscles, so both your skeletal and your cardiac, so your heart muscle, as well as your brain and your retina and sperm. And so, but the muscle is your the main user of this creatine. So about 94% of your body's creatine is located in your muscles. And so it's really, really important for the muscles. So creatine is able to act as a sort of buffer. So it's able to help So creatine is able to act as a sort of like phosphate buffer. So we talked about buffers usually in biochemistry. If we're talking in a buffer, we're usually talking about like a pH buffer. So something that's giving and taking protons in order to stabilize the pH, which is a measure of proton availability. But we can buffer other things. And so creatine is buffering these phosphate groups. And so it's buffering like the ratio of ATP to ADP. But remember, like creatine, not all of your cells have creatine, are using like creatine like this. So yeah, it works great in your muscles, but what about your other cells? So it turns out that ATP and ADP can be regulated. The levels of them can be regulated in other ways as well. And their levels can also help regulate other things. And so the ratio of ATP and ADP is tightly coupled to metabolism. So metabolism is the making and breaking of molecules. So it involves anabolism, which is making molecules, and catabolism, which is breaking down molecules. So if you think about what's going to happen, if you have a lot of ATP, then you're going to want to make stuff, right? And so when you're making stuff, then you're going to be decreasing the amounts of ADP and making more ADP. You have a lot of ADP, now you're not going to want to make stuff, or at least you're going to want to get more ATP before you can make more stuff. And so you're going to need to do catabolism. If you have ADP, you're going to need to break down more ATP. So if you have a lot of ATP, now your cells are like, okay, dude, like hold off a minute. don't break things down. I want to use those pieces instead of breaking things down. I want to use like some of those pieces. A lot of times, because catabolism and anabolism, like the pathways are like these webs and things can go other directions in a lot of different places. And so if you keep breaking these molecules down, you can break it down all the way, get the most energy. But now you're going to have to remake those like intermediate pieces from scratch. And so you don't want to do that. And so what's going to happen is that if you have a lot of ATP, you want your cells to stop breaking things down, at least for a bit, while you can use up the ATP you have. And so when this is going, ATP is going to allosterically inhibit glycolysis in the citric acid cycle. So glycolysis is where you're breaking down like a sugar. And then the citric acid cycle is the part that's after it. And then that goes into oxidative phosphorylation, the electron transport chain, which is where you get the big payoff in ATP. But the important point is that ATP is going to inhibit those. So glycolysis in the citric acid cycle are when you're breaking things down, and ATP is going to inhibit the enzymes that are breaking things down. It's going to do this allosterically. And this just means that the ATP isn't binding like in the active site. It's not binding, it's not competing directly for the substrates of those enzymes. So it's not like binding in the active site where the things that get broken down bind. It's binding somewhere else, but it's changing the shape of the enzyme so that it's not going to either it won't bind those other things or it won't be able to process them efficiently. So the ATP, when you have a bunch of ATP, then it's going to bind to those enzymes in a way that inhibits them. So you're going to stop breaking things down, but then you're going to start using up the ATP and getting more ADP. So when you have less ATP, then this inhibition is going to be released. And so when you have this inhibition released, because you don't have a bunch of ATP around that's binding, because this binding is like reversible and stuff. So it's not like you're like using up all the ATP and binding to the enzymes. But so you then relieve this inhibition. And you want to start, now you have a bunch of ADP, and you want to start breaking things down, which is good because you've relieved this. And also the ADP can act as an allosteric activator of those processes that ATP was inhibiting. So it's allosterically, so it's not in the active site again, but it's instead of inhibiting it, it's activating it. And so now you're activating these things that you want to activate when you want to break things down to get ATP. So you have this regulatory feedback loop that's where you have ATP regulating metabolism and metabolism regulating ATP. And so everything is kept happy. And so, yeah, so basically your bodies have different ways in order to regulate ATP and ADP levels. And your muscle cells have this creatine phosphate pathway and that they can use so that they don't have to wait around for the longer pathways and that sort of thing. And so your muscles can kind of make ATP on demand.